Good morning, everyone. Welcome to the NanoBio4Can Webinar Series. First, I'd like to give a brief information about the project. NanoBio4Can project, a mobility scholarship program for the postdoctoral researcher, aims to develop nanobiotechnologies for innovative therapeutic approaches in cancer. While Sunum is the coordinator of the project, İzmir Biomedicine and Genome Center, Koç University Research Center for Translation Medicine, and TÜBİTAK MAM are the implementing organizations. For detailed information, please visit nanobio4can.net. Today, we are kicking off the series with our first webinar. We have Dr. Ankush Sontakke from KUTTAM, Dr. Ika Nurlaila from TÜBİTAK MAM, Dr. Fazel Gorjipour from İBG. Each researcher will speak 15 minutes, followed by 5-minute Q&A session. We will organize this monthly webinar within the framework of the MSCA COFUND Grant Agreement Dissemination and Communication Obligations. This webinar will be recorded. It may be posted on YouTube. If you do not consent, you may keep your camera off. Dear all, welcome once again to the webinar. Thank you for your valuable contributions. We will start with Dr. Ankush. This stage is yours. Would you introduce yourself to us while starting the webinar?
Good morning, everyone. My name is Ankush Sontakke. I am working as a MSCA COFUND postdoctoral fellow under the NanoBio4Can program at KUTTAM, which is Koç University Research Center for the Translational Medicine. I'm working with Professor Funda Ajar. She is the professor in the Department of Chemistry, and she is working as a Vice President for Academic Affairs. I would like to start introduce you all with my academic and professional background. My all academic journey I completed mostly in India in various institutes. It started with my undergraduate program in chemical engineering from the Jawaharlal Nehru Institute of Engineering and Technology, and my master's from this one of the central university called Sant Longwal Institute of Engineering and Technology, which is in Punjab. And I completed my PhD from one of the premium institute in India called Indian Institute of Technology, Guwahati, in chemical engineering department under the supervision of Professor Mihir Kumar Purkait. As I told, I am working in Koç University as a NanoBio4Can postdoctoral fellow. I have started my journey here in last May. Prior to that, I was working as a tenure faculty in Technical and Applied Chemistry Department at VGTI Mumbai. During my PhD, I have also worked as a teaching assistant for the Central Instrumentation Facilities and the Department of Chemical Engineering at IIT Guwahati. Prior to join my PhD program, I worked as an assistant professor in the Department of Chemical Engineering in Chandigarh University. And prior to join my master's program, I worked in one of the chemical industry called Multi Organic Private Limited as a graduate training engineer. In terms of my research background, which mostly aligns with the structural, functional, and morphological modifications of nanomaterials, I am mostly dealing with the nanomaterials related to graphene and metal organic frameworks for their application in novel drug delivery systems, including the cancer therapeutics and targeted drug delivery systems. Now here in NanoBio4Can program, as a postdoctoral fellow, I mostly deal with the photodynamic therapy related applications. Also, I have worked with the wastewater treatment related applications during my PhD. In today's webinar, I will be presenting my work related to sustainable metal organic frameworks as a targeted drug delivery systems for photodynamic therapy. In this webinar, I will be discussing about the background and motivation of my study and the challenges and the scope of the study related to that. I will be concluding my webinar with the overall objective on which I'm working and some dissemination activities which we are conducting here under the NanoBio4Can program. To start with the background and motivation, I would like to start with cancer. So cancer is basically not any foreign particle coming to your body. These are normal cells which are growing in abnormal manner, which are tends to escalate in an uncontrolled way and which provides like higher mortality rate amongst the victim in the world. If you're talking about the treatments or the methods to treat cancer, there are various one. But the chemotherapy is the most common one, where we are using some anti-cancerous drugs which are toxic in nature, which are used to diminish or halt the growth of weakly isolating or rapidly growing cancer cells in the human body. These chemotherapeutic drugs also come up with some challenges. These drugs are basically unable to distinguish between the normal healthy cells and the tumor cells, which causes due to its non-specific targeting. And the rapid use of these chemotherapeutic drugs uh, leads to immunity for cancer cells and provides the multi-drug resistance as well. And the rapid use also provides some adverse impacts while generating the cumulative toxicity for various organs in human body. So researchers come up with various remedies for these challenges, like to use the nanomaterial assisted drug delivery systems and the controlled and sustained release of chemotherapeutic agents, with various advantages including their smaller dimension and stimuli responsive behavior. If we are talking about the nanomaterial assisted drug delivery systems, which also possesses various challenges, these nanomaterials are mostly poor in tumor penetration and provides heterogeneous EPR, that is enhanced permeation and retention effect, and it also has some non-specific targeting behaviors. And the most important thing is it may cause the premature or uncontrolled release of the drugs. These challenges can be overcome with some advanced remedies by using these nanomaterials for PDT application, PDT, that is photodynamic therapy related applications, the targeted drug delivery approach, and the combination of both these therapies, with some advantages like with having enhanced targeted approach and the smaller dimension of these nanomaterials provides better biodistribution when we are actually implementing this for cancer treatment. And these nanomaterials can be functionalized with some near IR active materials to provide deeper tissue penetration. And the most important thing again is the stimuli responsive activation and release of photosensitizers or drug within the cancer cells. As we are talking about photodynamic therapy, let me start with introducing the photodynamic therapy here. It is basically a non-invasive technique which has minimum side effects through their targeted approach, where we are actually using some photosensitizers. Those are the materials which get activated when we irradiate with some specific lights. Once these photosensitizers are activated, they will generate some free radicals and singlet oxygens to via type 1 and type 2 electron transfer to kill the cancer cells. And in today's world, there are various photosensitizers are already in the clinical use, like 5-ALA and chlorine E6. But these photosensitizers itself has various drawbacks. Like this can cause some fluorescence quenching, and the aqueous stability of these materials or these photosensitizers is quite a concern. And if you are talking about PDT requires oxygen for its better therapeutic efficacy, and the cancer environment is more of like hypoxia. It suffers with hypoxia, and also the higher level of GHS, that is glutathione, which is a natural antioxidant, will also affect the overall therapeutic efficacy of the cancer cells for cancer treatment. And another thing is the limited tissue penetration. In general, now in clinics, in medical sciences, we are using blue light emissions to get rid of cancer cells, and the blue light does not have much deeper penetration, so it is unable to treat the cancer cells which are present into depth of our body. So. If we are talking about if we are considering the targeted drug delivery systems, which is basically we are using some chemotherapeutic agents to site specific release of this agent to control the overall toxicity of these drugs. It involves some nanomaterials and antibodies for providing more targeted approach, and that will leads to better drug distribution and overall enhancing the therapeutic impact of these drugs. As we can see in this figure, these nanomaterials can be functionalized with the targeted ligand to provide better endocytosis with enhanced permeation and retention effect, and release the drug or chemotherapeutic agent to the targeted site. There are various types of nanomaterials are currently in use for such applications. I am mostly focusing over the metal organic frameworks. The metal organic frameworks are basically the crystalline compounds which consist of various organic molecules which are held together with the help with the metal ions. These are having higher surface area and the porous morphology. And the most important characteristics of these MOFs are they are more enriched of organic molecules, so it can be functionalized further on for various applications, for including the wastewater treatment and targeted drug delivery systems as well. This functionalization ability will be used to have better targeting for cancer cells. Uh, and if you are considering MOF for the targeted drug delivery related application or even photodynamic therapy, it also suffers with some challenges. The synthesis method currently used for the synthesis of MOF are mostly the conventional methods include hydrothermal or sormothermal synthesis, even the microwave radiation. These processes involves high energy intensive protocols and some harmful chemicals that makes this synthesis more costly and environmentally unsustainable. So another thing is if you are considering for biomedical related applications, we should the larger particle size of MOPS is also a concern. So in order to get rid of with all these challenges, we can have some remedies, like we can adopt a sustainable synthesis strategy by using water as a solvent and um, room temperature condition to synthesize our MOPs. And as I discussed, like we have more organic molecules present on the MOPs, so we can functionalize these MOPs with the help of many targeting ligands as well as drugs, photosensitizers, and with another materials with higher loading capacity. And the larger particle size of the MOPs can be get rid of by varying the reaction parameters during the synthesis of MOPs. So in line with that, I'm as a NanoBio4Can postdoctoral fellow, I'm working with here at
Ankush, 5 minutes.
yeah, synthesis and engineering of sustainable redox active nanomorphs and their composite for enhanced targetability and stability and photodynamic effect. Herein, we are synthesizing the nanomorphs to provide more synergistic approach to get rid of GHS level within the cancer cells and to release the photosensitizers to provide more ROH generation, and that will certainly kills the cancer cells. Uh, yeah, in this work, till now we already have synthesized the nanomorphs with the average size of around 80 nanometers, and we have successfully employed it for the PDT related applications, and they have provided better results for in vivo PDT effect as well. Sorry, in vitro PDT effects results as well. Under the NanoBio4Can program, we are actively involved in various dissemination activities. Previously, like in last year, I have attended one of the nanoscience and nanotechnology conference called NanoTR in Ankara, where our work has been awarded for the best poster prize. We are also actively involved in some public talks and trainings. Specifically, like last 1 month, we have given a talk for high school students where we have discussed about our work in a most easiest way so that the high school students should understand. While working in the field of targeted drug delivery systems and nanomaterials for like around 7-8 years, I have published one book for the in the same area and various research articles, and some are already in the pipeline. At the end, I would like to thank the NanoBio4Can program and the funding agency like such as EU, European Union and TÜBİTAK for providing me this opportunity. And I will also thank the host and implementing institute like Koch University, KUTTAM and Sunu for providing me all the infrastructure and facilities to conduct my research here at Koch University. At the end, I must thank to my mentor here in Kochi University, Professor Punday Ajar, and my Funtech family for their continuous support and guidance through during my research here. Thank you so much for your attention. I will be happy to have questions hereafter.
Thank you, Dr. Ankush. Q&A session is open now. You can just speak up or you can use chat box. Yes, Mustafa.
Hi. Thank you so much for Ankush for this nice presentation. I actually have two questions. Like the first question is actually regarding the difference between the metal organic framework and also the silica. So I mean, both of them actually have porous structure. Like the silica is actually like if we're talking about the health concerns and the safety measurements. So I mean, a lot of studies have been talking about the silica, and this is biocompatible. So I have a question regarding the safety consideration for the MOFs. This is one thing. Second thing is actually about the size of the MOFs. So some of them actually, like I see some of the polydisperse sizes. I mean, with the your SEM
PDI.
image that you provided. So could we have like any like like any parameter that you could control the size of these MOFs, because like the size matters a lot. So when you're talking about the endosomal escape or even like the penetration, so I think like the thing need to be like addressed. Thank you.
Yeah, thank you so much. Yeah, the first thing is like about size. You asked like the size of the MOFs actually can be controlled by controlling the growth rate and the nucleation rate of during the reaction. The nucleation rate can be controlled by various means. If we like, I can give you an example. Like if we can increase the stirring rate, the nucleation rate will be higher. And like we use some modulators and surfactants to control the size of the MOFs. If you're talking about the biosafety level of this MOF, still now there is no such study already been conducted specifically for biosafety related things. But yeah, as it is variable in size, so we are trying to make it in a uniform size with better polydispersity index. And if the size is lesser in a nano range, then the biodistribution will be better way in a better manner, and that will certainly improve its bioavailability and biocompatibility for our application.
Yes, Hasan.
Thank you for the nice presentation. I have a small question. You said you are going to use them for photodynamic therapy. Maybe I missed that part. But what kind of light or photodynamics are you going to use to utilize these structures?
We are trying to utilize the near infrared region, red light actually, for this application.
You mean infrared or red? Yeah, I mean like
No,
800
near
nanometers or
near IR.
near IR. Okay.
Yeah.
Okay. Thank you very much.
Thank you.
Do you have any question?
May I, Dr. Hojam? Dr. Ankus, that is also small questions. I think, maybe I missed it. If that's reached that clinical implementation, so do you think this prototype can be used for blood cancer, which is fluid or solid one?
Fluid or solid? Can you please repeat that part?
If this reach clinical implementation later, because we all want that, do you think it what is it? It can be used for what kind of cancer, whether it's fluid like blood cancer or that solid one. So it has localizations, fixed localizations.
Yeah, we are first trying to have more in liquid phase. What I understand your question is based on that, like we are trying with the liquid phase, and yeah, we are trying to make it more aqueous soluble so that it can be injected within the like for the human body, you can say.
Clear enough, Dr. Ankush. Thank you.
Thank you, Ika.
Dr. Ankush, thank you for
Yeah.
your presentation.
Thank you, ma'am.
Dear participants, thank you for your contribution. Yes, Dr. Ika, the stage is yours.
I'll stop sharing then. Thank you so much.
Yes, please. Thank you.
Sure. Thank you.
Ika, you are muted.
Right. Merhaba herkese. So I'm Ika Nurlaila. I'm very excited to share a glimpse of my work under the NanoBio4Can Medicinal Discovery Action 2020-2025 program. Right about the title that strongly indicated there. In simple terms, my research focuses on something called antigen processing machinery, or shortened as APM. You can think of APM as cell's internal system that prepares and presents signal in the form of tiny peptide fragments that tell our immune system whether something is normal or dangerous, like cancers. Why does this matter? At least for me, because even though cancer cells are abnormal, they often find ways, always find ways, not often, always find ways to hide from immune system. What we are trying to do under this program is to better understand and improve this antigen processing machinery system, so we can retrain the immune system to better recognize and attack cancer more effectively, more efficiently. So today I'll walk you through how I studied this system and why it could open new doors for more targeted and effective cancer treatment. If we look at the anti-cancer drug market, the growth is striking. In Turkey, for instance, the market was about 1.5 billion USD, which is a lot, in 2022, and is projected to reach 2.8 billion by 2030, with the compound annual growth rate of 8.9%. And it is similar story in Indonesia, even with a higher COG. So what does this tell us? This tells us that cancer is not only a major health burden, it's also driving a rapidly expanding therapeutic landscape. But despite all this growth and investment, many current treatments still face limitation as of resistance, relapse, and in some cases, patients simply don't respond at all. This is exactly where the need of next generation targets comes in. Instead of only focusing on killing cancer directly, we need to rethink of how cancer is seen by immune system, and that bring us back to antigen processing machinery. Antigen processing machinery sits upstream. It determines whether tumor antigen are properly processed and then presented to immune cell. And if this system is impaired, even most advanced immunotherapy in the world may fail, because immune system simply don't see cancer as a cancer. This is friend, that's a part of the body. So by targeting APM, we are not, I'm not only just adding another therapy, I potentially unlocking the effectiveness of existing one. That's why I believe that APM represents a strong candidate for next generation of cancer immunotherapy. So Here about me, I'm permanent researcher at the National Research and Innovation Agency, also known as BRIN in Indonesia. Our head office in Jakarta, if you ever been there. It's really privilege for me to be part of this growing and keep growing scientific organization owned by government, and I'm proud to represent BRIN here, where we are committed to advancing research that has real impact for society. So in case everyone here got something in mind to collaborate about in next future, you at least have me, so please contact me. My journey in science has been driven by very personal and professional interest in cancer immunotherapy. For me, cancer is not just about understanding disease mechanism. It's about ways to translate that knowledge into solutions that are accessible. That's the most important thing in Indonesia, because we actually have a lot of cancer treatment, cancer platform. Everyone knows about it, but not everyone has access about it because of economical constraint. That's scalable and meaningful for patients. That's our long-term homework. So that's my one of long-term goal is to contribute to the development of more generic or broadly applicable immunotherapy. And I got my education, the latest one, from University of Birmingham, where but actually my first sight on cancer immunology is when I did my master in University of Brunei Darussalam, because that the first time I learned specifically, the only one every day learn about cancer immunology. Yeah, there you go. That's my base. So that's the umbrella. It's like TÜBİTAK, that's the umbrella. But I specifically joined at Center for Vaccine and Drug, where related with how to develop vaccines, how to develop adjuvant for drug delivery, new drug development, like that, everything centered in my research centers. Now, let's talk about what is APM, why APM is so important for me. For this, we can have a look. Everyone has it in one single cell of ours, so this machine works every day. So the protein are processed into peptide and properly presented on MSC class 1 molecule for recognition by cytotoxic T cells. That's what we want. It starts with EROT degradations, which identified misfolded or unwanted protein and retro-translocate them from the endoplasmic reticulum back to its cytosol for degradations. These proteins are then broken down into smaller peptide fragments, and then these peptides are transported back into the ER by TOP1 or TOP2, which acts as a selective gateway to ensure that only suitable peptides enter the ER for presentations. The other will be degraded soon. Once they reside back in the ER, a coordinated chaperone system takes over. Being connexin will stabilize newly synthesized MHC class 2, and then calreticulin and BIP binding immunoglobulin protein and other chaperone assist in the proper folding. That's chaperone being heat shock protein also to maintain the structural integrity. And a critical component here is stepsin, as you can have a look here, which act as a bridge between TOP and MSC class 1. This functions to optimize peptide loading, and in parallel, 61 translocon 61 contribute to antigen processing by allowing protein movement across the ER membrane. Sorry, I'm having cough. So altogether, this coordinated network make sure that antigenic peptides are efficiently processed. What if not? Meaning that what APM do is very key, because it is not only, if we talk about the antigen, it's not only about presentation, but in clinical realm, it will determine resistance and also irresponsiveness. That's what I'm keen to do for. And inspired by a publication by Jeffrey Thompson, 2020, APM was found to have a predictive value for non-small cell cancers. If you have a look here, this significant gene signature consists of this gene. It can stratify a high risk of NSCLC. With that, I question how this defect affect antigen presentations. So meaning if there is defect, the repair for this defect will improve antigen presentation, so that we can expect that antigen can be present very properly, and then the T cell will be more sensitive to capture them, and then from immature T cell, they got matured and differentiate into these different subset. I mean,
Ika, 5
to
minutes.
address that, okay, to address that, this APM defect, APM defect detected in blood. And yes, I did the metadata analysis, and if you have a look, there are genes in the APM detected from metadata, meaning I extracted a lot of gene data set from GAO, and then yes, we found the APM gene over there. And this complex set, the finding from the previous one actually come from different units. That's come from proteasome transporter and so on to MSC. With the addition of translocon 6 with this kind of variant, meaning that antigen processing machinery exists in cross carcinoma, thus urge further study. And this is also evidence, supporting evidence from original research that I did from my previous work back there in Indonesia. Actually, I'm in Indonesia. Back then in Indonesia, we're using bulk RNA sequencing, but with breast cancer tissues, it appeared that yes, APM gene, not all of course, some of APM gene appear to be determinative and distinguishable between those tumor triple negative breast cancer with healthy tissue. So meaning what? Multiple gene from APA complex appear for cancer stratifications. And the next one, that's what I did recently, not all. This show that cancers will compromise the immune cell subsets. This what panel that I use, it result from my strategy pretty much aberrant, because I didn't add a live death, but I play around with where the singlet located in this gating, and this result in some dynamic either in CD3 negative population and CD3 positive population, with the dynamic is clearly seen over here, but I didn't put any asterisk statistical mark, because we still gather more healthy donors. This might change the dynamic, but overall, this is what I can get so far. With that, I can conclude that, but with continuation, because the project is still ongoing, the dynamic of immune cell compositions and transcriptomic landscape constitute the robust tool for informing the engineering of downstream immune response. Most likely, nanobiotechnology can also benefit from these informations. which are fundamentally initiated and driven by immunogenic antigens. I think that's less than 5 minutes, Dr. Hojam. So I return the screen to you, Dr. Hojam. Thank you so much. So I'm waiting for any feedback or in terms of research or any other potentials.
Thank you, Ika.
Thank you a lot.
Yes, now this session is open for questions. You can just speak up or raise hand, or if you like, you can use chat box. We have 5 minutes. Fahd, yes, Fahd.
Thank you, John. Thank you, Dr. Ika, for the presentation. I have a question that might be a bit off the scope of your presentation, but
Okay.
as far as I understand, we do have some endogenous cytokines that sort of modulate the APM landscape, like interferons
Yeah.
and TNF-alpha.
Yeah.
So in your experience, are there specific cancer types that mechanistically are more suspected to escape the immune system through these APM mechanisms, and that would be like, low-hanging fruits for, APM modulation immunotherapy?
So answer no. Thank you, Dr. Khalid. About the APM, to my perspective, there are a lot of contradictory, but not contradictory actually, a lot of perspective on this APM. But to my view, that APM would be more distinguishable in solid cancers, because why? The tumor microenvironment play a much bigger role than the blood cancers, because there is a direct contact between the cancer and lymphocyte, infiltrating lymphocytes. So they play around with that, and the defect that says one gene that got defect, and that will affect the downstream genes later on. So once defect might trigger another defect in downstream of the genes. So that's what I'm thinking. So the modulation, the reinvigoration using the cytokines modulation, they might affect but superficially, because say if the seed doesn't appear or disappear but with mutation, so that the function is silenced, even though you will add tumor necrotic alpha or interferon gamma, which just work very well in vitro, but that in solid cancer they have interaction with that tumor microenvironments. So that cytokine might work, but in the counts of hours or in short time. But after that, the seed is not there. So no translocon that will allow this process antigens to be endogenous process. No, sorry, extracellular antigen to be exposed to CTL or CD8. So if no such aspect, no such variable that help the antigen to be exposed, even though there are surveying lymphocyte around the cancer cells, they won't recognize. Maybe the modulations work, but temporary. But still, we need to work on the machine. The manufacturer doesn't work, so that's why we need to fix that. But then, the black hole is, we don't know what is the major defect in that. Whether this work only in superficially, whether it's in epigenetically levels or in genetic levels. or things like that. So a lot of black holes in APM. So I think that's what makes APM interesting to me, captivating to me. So hope I answered, Dr. Bud.
Yes, thank you.
Any questions? We have 1 minute. Dear Ika, thank you for your presentation.
Thank you, Dr. John.
Thank you. Dr. Fazel, are you ready?
Hello. Yes.
I stop my screen share and you can share your screen.
Okay.
Stage is yours.
Thank you. Hello, everyone, and thank you very much for giving me this opportunity to present my work here. My name is Fazel Gorjipour, and let me. Do you see the slides in full screen mode?
Yes.
Okay. Let's get into my background in education. I have PhD in biomedical sciences with focus on applied cell sciences from Iran University of Medical Sciences in 2019, and Master of Science in Medical Biotechnology from Iran University of Medical Sciences. Actually, I've been almost 20 years in the same university, having all my education, academic education and career at IUMS before joining the IBG at Izmir. I'm currently working as a MSCA COFUND postdoctoral fellow at Izmir Biomedicine and Genome Center in Izmir, where I'm researching the use of antibody functionalized liposomes for targeting malignant pleural mesothelioma. Before this, I was working at Iran University of Medical Sciences as a senior researcher, where my focus was on material science and protein chemistry and immunochemistry, and I have also keen interest in standardization of the science through evidence-based medicine and also evidence-based preclinical medicine. I have published some papers. Since I believe that conducting a science is something very important, but ability to critically appraise the existing data and and improving the conclusions and synthesizing new evidence from them is also important. However, let's get into the current project. Actually, I'm going to present here when I came to start my experience at Izmir Biomedicine and Genome Center. I was supposed to use this delivery system based on liposomes functionalized with antibodies for targeting malignant pleural mesothelioma, and it's when I started the project, it's opened a completely new venue of experiences to me. The project was to target the mesothelioma using a BOP1 kinase inhibitor, which was previously identified in our lab using CRISPR screening in MPM cells. The inhibitor was by 1816032 which was a quite novel molecule. There was actually there was not much information about it and its pharmacological behavior indeed. And we just knew that it's a very potent inhibitor of the BOOP1 enzyme, but the amount that was needed for inhibiting the growth of cancer cells was much more than what we see in enzyme inhibition assays. Despite being a potent drug, yet we needed much more higher doses. Then we actually found that targeting may be a key to successful use of this drug. We had the idea to actually formulate this as liposomes, but there was no data about methods to study, quantify this small molecule. And there was a few publications, only one citing a technique based on mass spectrometry, which was expensive and also was not available to us to use during our experiments. Then I decided to check for the spectral and photophysical data. There was no data again, and I started the characterization of the molecule. Here we see the spectral characteristics of the small molecule, and the absorption peak at 310 to 313 nanometers, and emission peak was around 360 to 364 nanometers, and we observed a moderate fluorescence with this molecule. Then we decided to use these characteristics for measuring the molecule which is to be used in formulation process. We set up the UV spectrophotometry study, also direct fluorescence assay, and these two techniques were conducted without extraction, and it gave a good sensitivity, about 1 micromolar, for measuring the molecule in the serum, which was lower than the IC50 we identified with in vitro cell culture studies. We also set up RP-HPLC with fluorescence detection, which we needed to extract the drug from the serum for HPLC, because the serum compartments interfere with the HPLC and may cause the actually plug in the HPLC column. Fluorescence measurement was more sensitive, while it needed larger volumes of the samples, but UV spectrophotometry using a nanodrop needed much less samples, which give us wide choices according to the application during the measurement of the drug concentration. During the HPLC experiments, we need to extract the protein, extract the drug from the serum and precipitate the proteins. Then we did protein precipitation using acetonitrile, and we did observe that signal is lost when we precipitate proteins. it appears that the drug is co-precipitated with the proteins, and we didn't have any drugs remaining in the fluid compartments, and it was all precipitated with the protein as a pellet. Then we thought that there should be a strong binding between the drug and the serum proteins. Do you have the picture on your screen?
We see unexpected observation in drug serum extraction.
Do you see this? Let me take it.
Stop and share again.
Okay.
Bir dakika ekleriz.
This is the process of sample extraction and recovery from, actually this was a spike and recovery process we used for purification of the drug from the serum samples. This one is the serum containing the drug added before starting the experiments. This one was used to see if we have lost the drug during the acetonitrile precipitation of the proteins or not, and this one was to control the drug loss during this actually filtering process, because the drug is a hydrophobic molecule and it may bind the filter and lost during the filtering process.
5 minutes.
We lost drug when we actually precipitated the proteins using ice cold acetonitrile, but after adding 20% of the MSO, we were able to recover the drug successfully. Let's get back to the slides. Okay. Successful recovery using 20% DMSO indicates that there is a non-covalent interaction with a strong affinity between the molecule and the drug. We have actually shared this the prepublished version of this work on SSRN. It's available as a prepublished preprint, and you can and everyone interested can access it through this information available at the bottom of this page, this slide. From this observation, we found that the drug is tightly bound to serum proteins, and since most of the protein, the major compartment of the serum proteins is albumin, we hypothesized that albumin may be the main binder to Y186032, and we wanted to see whether this may affect the activity of the drug. We used molecular docking, fluorescence quenching, and functional assays to find the interactions of the drug with the albumin. Then molecular docking reveals binding sites at pseudo site 1, which is a major binding site on albumin. Then with actually can say a somewhat strong binding. It was near to troponin 212, which is responsible for the fluorescence of the albumin. Even at low concentrations, the albumin, the drug was able to quench the albumin fluorescence, and this activity was increased under acidic conditions. We also tested the bioactivity of the small molecule Bay18-16032 on AB12 mesothelioma cells at the presence of increasing concentrations of the albumin, and observed that increasing BSA concentration decreased the biological activity of the drug against mesothelioma cells, AB12 mesothelioma cells indeed, and it caused the increase in IC50 of the drug. Then it makes the treatment less efficient. We found that albumin actually sequesters the drug. It's moderate to a strong binding affinity. It binds to the drug and reduce the free drug fraction available to be uptake by the cancer cells, and it limits the bioavailability in circulation indeed. Actually, the increased doses we need to inhibit the tumor growth in vivo is not due to failure of the drug. We think that it's actually a delivery problem, and could be improved. The results could be improved through a good delivery and targeting strategy. Overally, as we see here, under ideal scenario, drug is fully available in the serum. It reaches the target cells and induces apoptosis. But in reality, we see that drug bonds to the albumin. It's sequestered and can't reach the target cell. And the functional consequence is the loss of bioactivity and higher doses needed to actually treat the tumors in vivo. To overcome this, we are actively developing liposomal formulations targeted with antibodies to enable the drug to reach target cells and kill them. This is already underway, and we will share the data in future meetings. Thank you everyone for your attention. I would like take this opportunity to appreciate everyone, to say thank you to everyone who supported me during this research. Our great research team at Şentürk Lab in IBG, also the NanoBio4Can Program, MSCA and TÜBİTAK for funding of the project, and also NanoBio4Can fellows and team members and other collaborators who actually supported me during the whole work.
Thank you, Dr. Fazel. Q&A session is now open. You can just speak up or raise your hand, or you can write on the chat box. Yes, Hasan.
Fazel, I have a small question. You said you are thinking it's binding to albumin. I mean, you are pretty sure about it, but is it reversible, or is just like binding and completely losing its efficacy and maybe a conformational change?
Actually, we tested in different settings. We used fluorescence quenching. The drug was binding to the molecule with much strength that it quite suppresses the fluorescence from albumin. Then it hints a strong static binding. Then we expect it, to be static and not, and the drug was not released under normal conditions. We were able to release the drug from the protein fraction from albumin only in the presence of 20% DMSO. We also did the docking studies, which showed that the drug was adjacent to the residues with some actually points to be close to residues from the albumin less than 4 angstrom, which also implies a very tight static binding again. We have several confirmations for this, and also in the cell culture, we also saw the loss of activity or decrease, at least in the activity of the drug, actually proportionate with the concentration of albumin in the cell culture medium. Then every evidence that we had achieved during a study confirms the tight binding of the drug to the albumin and inhibiting it's actually sequestering, let's say, sequestering the drug and decreasing its efficacy.
Okay. Are you planning actually to modify the drug? So you can actually inhibit the interaction between albumin?
I would like to share is that we also tested the release of the drug, or its binding to the albumin, how it changed in the with the pH, with the changes in the pH of the medium, with the pH going down to almost 7 or even less. We had tighter binding, which was confirmed with the further reduction of the fluorescence level of the albumin. Then we see that there is a decrease of pH in the tumor microenvironment, which further promotes this delivery problem. Since the drug is not released from albumin, it binds tighter to the protein in a lower pH. We expect it not to get released easily from the molecule in tumor microenvironments. And it further actually contracts the activity of the molecule.
Makes sense. Thank you. Thank you very much.
Yes, Fatih.
Thank you. Thank you for the presentation, Dr. Fazel. I do have 2 questions, actually 1 question and a comment. First is as a follow-up to Hassan Hoja's question is, you did mention that based on your assays, your fluorescence measurements, you noticed, you found tight binding between the drug and albumin. But when it comes to pharmacokinetics, your conclusion was that eventually this causes an increase in the effective dose, the IC50 of the drug, because most of it is like bound to albumin and taken out of, it's not available, basically. But do we know the time scale for these interactions, right? So do we know how fast, or if at all, this drug eventually dissociates from the complex? Have you considered making dynamic assays like with biolayer interferometry or SPR assays that can actually tell us if this drug, when it is bound to albumin, how long it takes to dissociate from albumin? Because when we go to the clinical picture and the drug is taken and it is sequestered by albumin, it is possible that after a set amount of time, that drug is actually released and then there's a spike in serum concentration. So I think these are insights that you might also find helpful when talking about the pharmacokinetics of the drug. In these free states before, encapsulating it in liposomes and other approaches. My second comment is about the image you showed for extracting the drug from serum. So it appears that, in one of the approaches, you recorded about 101% concentration of the drug, and then you have 100% and we have about 87%. I am assuming that these measurements were actually taken in replicates, right? So it might be helpful to include, variance calculations so we can actually see that the effects of these different steps in the purification are actually statistically not affecting, the amount of drug recovered. That will be all from me. Thank you once again.
Yeah, thank you. About actually the length of the or duration of the drug binding to albumin and its release from the albumin again. We didn't do an experiment, especially SPR. It's available at our institute, but the budget didn't let me to do that, and actually it wasn't the main part of my work. However, I followed up the efficacy of the drug. We do this IC50 studies for 72 hours after the addition of the drug, and sometimes we can even keep it more longer, for example, for 4 or 5 days or even more. We did it in for some durations and didn't see any change in the actually activity of the drug. And for us, activity is the main target. Therefore, we just considered it enough for this part of the work. And yeah, I'm personally interested in studying that interaction using SPR, and I will do it if there is an opportunity. And we could secure funding for that kind of studies, yeah. But the activity, just to wrap up, I should say the activity remained the same. And since the observed binding is of static type, according to all evidence that we have, we think that the binding is, some kind of that's not dynamic, the static binding. We have the static binding versus dynamic binding. In case of a static binding, it's not released easily. And I personally don't think that there should be such spike in release of the drug from albumin.
Thank you. Thank you, Fazel. Thank you for your presentation. And thank you, Dr. Ankush Sontakke. Thank you, Dr. Ika Nurlaila. And thank you again, Dr. Fazel Gorjipour, for your presentation. And thank you all participants for your participation. The second
Thank you.
webinar in our series will be held on
Thank you.
April 30. Dr. Kashif Maroof, Dr. Melike Belenli Gümüş, Dr. Mina Masoudi will present information about their research. Until then, goodbye.
Thank you to all.
Thank you. Thank you
Bye-bye.
so much.
Thank you.
Görüşürüz herkese.
Görüşürüz İka. Hoşça kal.
Görüşürüz.
İyi günler herkese.
İyi günler Fazel.
Sağ olun.